Actin assembly alone can do useful work to deform the cell membrane, for example, even to explain a lot of aspects of complex behaviors like cell crawling. But even cell crawling has a few more subtleties that it requires than simply assembly. Of course, it requires disassembly as well. So the large universe of actin binding proteins greatly expands the repertoire of what cells can achieve with the cytoskeletal polymer, given, the starting material, a bunch of actin filaments. Well, of course, in the context of cell crawling, we already mentioned, one of the things you could do is chop them up, which perhaps you might need to do if you need to pick up the rear of the cell or something like that. Of the separating proteins that are important here, I already mentioned cofilin. Another one commonly encountered is gelsolin, which happens to be a calcium-sensitive actin severing protein. So that's one thing you can do. We also already mentioned motors. Whoops, that's not going to work, is it? Myosin only crawls towards barbed ends, but let's say we have a more constructive configuration, right? Motors can engage with filaments that are properly oriented, and they can mediate contraction so that, for example, loose networks might become over time tighter bundles. You have to still put the myosin in there. Now, cross-linking proteins without motor activity could turn this spaghetti plate of filaments into a gel. One that I'll mention is the protein filamin, which has two actin binding domains linked by a relatively flexible hinge. Filamin can assemble a group of filaments into the kind of loose mesh that you might expect to find, well, really anywhere that the cell assembles actin for the purpose of providing, say, the cortex with tensile strength. Some cross-linking proteins are more structurally demanding and make bundles instead of loose gels. An example is alpha-actinin. Alpha-actinin, let's try another color for it. Alpha-actinin also has two heads, but the heads are so configured that it makes cross-links amongst filaments with a relatively regular spacing, like so. Okay, let's turn that mistake into a teachable moment. Alpha-actinin cannot stretch across. It has a certain limiting length. So let's say it's not going to connect these two that are too far away unless, of course, some motion brings them within striking distance, in which case alpha-actinin will establish a cross-link and rectify that excursion. Another interesting bundling protein, another color here, fibrin, makes even tighter bundles, tucking itself amongst actin filaments. something like this. Those are relatively static. Here's an example of a somewhat more dynamic protein, the protein villain. Again, two heads. Villain is calcium sensitive as it happens. Let me write some names down here. This is alpha actinin. This is fimbrin. This is villain, not like the bad guy in the story, but villain as in microvilli. Villain also makes tight bundles out of these actin filaments with the property that in response to calcium, villain will not only let go, but it will actually sever actin filaments. So villain, found in microvilli, is both the agent of their structural stability and of their demise in response to some sort of signal that causes the cell to change shape by retracting its microvilli. There is, of course, a much larger repertoire of actin-binding proteins. These are representative ones that show up so often that they're worth getting to know by name, at least for the sake of this course. I should mention the lamellipod that we already talked about is based on the continuous polarized assembly of a branched actin network, which of course also has crosslinks within it besides the branches and is disassembled at its rear by severing proteins like cofilin in particular that prefer, as you will, aged actin, that is, actin that has already hydrolyzed its bound nucleotide. There's also a role for motors in the contraction of actin filaments remaining after severing toward the rear of the lamellipod. All of these things work together to make the kinds of dynamic structures that mediate cell shape change and make cells move. I also want to mention, in contrast to microvilli, which are relatively static cell surface projections that are based on non-contractile bundles of actin stabilized by crosslinkers like villain. Cells also express philopodia. If the lamellipodium is a sheet foot, phylopodium is think of it as a filament foot. A phylopodium is a long, sticky outy protrusion in contrast to the sheet-like lamellipod. Unlike microvilli, philopodia are often contractile. That is, they can reach out and grab onto something and then pull the cell toward it. Of course, in order to do that, they need motors, and you can't just put all the plus ends pointing outward then. In order for motors to engage within a phylopodium, they have to have filaments of mixed polarities. Finally, before we leave this particular family diagram, I want to point out an interesting consequence of severing. Let's say this group of actin filaments is the starting material. There are 4 barbed ends in there, 4 plus ends that actin monomers can actively add to. One immediate consequence of severing is that there are a lot more barbed ends. Whoops, I got this wrong relative to my original diagram. That was supposed to be here, here, and here. So point is, severing, in addition to perhaps getting rid of networks and loosening crosslinks, severing also potentially creates greatly more opportunities for assembly should there be sufficient monomer available to drive it. Now, let's go back to the motors over here. What happens if you just get a bunch of filaments and sprinkle them with motors? Believe it or not, this is an active area of research. Okay, so add motors to this. Excuse me, that's an unnecessarily sloppy arrow. Let me draw that a little more clearly. So add motors into this mix, and what are you going to get out? Well, okay, surely you could imagine that, I don't know, how many filaments did I draw in there? Are the motors all going to just randomly pull them into a smaller little wad? Or could motors order them? Now, let's assume that there are mixed polarities so that motors can actually engage. Under what circumstances will motors engage in such a way as to align the plate of spaghetti into some sort of ordered superstructure like that? Intuitively, the following argument seems to make sense. Motor engagement within this plate of spaghetti will achieve contraction to a point where the crosslinks within the network restrain it from contracting further. So they might do something like this, but likely it's going to stop at a certain point, at some point where you can't pull everything together tighter because there's just too many things that you'd be pulling against, too many other crosslinks. Some of those might be links to the rest of the world, of course. And intuitively, to proceed, one might imagine that if some members of this plate of spaghetti are tacked down, that weaving together a bunch of filaments through the action of contractility, mediated by motors, that is, might weave a fiber between attachment points. So again, intuitively, the density of crosslinks and the presence of attachments to the external world, whatever it is, the cell membrane, the nucleus, a microtubule, or a link to the outside world that spans the membrane, are going to determine the consequences, the organizational consequences of applying force to a bunch of actin filaments. Cells in the environment don't necessarily encounter a wholly uniform substrate, of course, and they don't make uniform contacts when they do. As the lamellipod advances, receptors for the extracellular matrix, whatever it is, tend to cluster in patches, small patches, sometimes fairly large patches. Now, I'm not drawing too many up at the leading edge because that's where the cell has just stuck its foot out. There might be a lot of incipient little clusters right behind where it's making new protrusions, and much larger ones further back. Such clusters are typically maturing into what are called focal adhesions. Pretty self-explanatory term if there ever was one in cell biology. And if you look at one up close, what you would see is something like this. If this is the plasma membrane, the focal adhesions consist of some sort of membrane-spanning proteins. Ugh. There, that's better. That have the means to link to the cytoskeleton on the cytoplasmic side. A large family of so-called integrins happens to be the basis for many such superstructures. In making links to the outside world to actin filaments, of course, there are other proteins involved. It's not just the integrins. There are bundling proteins, things that interface between the integrins and the actin filaments. And there might be other actin filaments in here too. Typically, focal adhesions become sites of attachment for cables of cytoskeletal filaments within the cytoplasm. And they are connecting in turn to extracellular matrix materials, collagen, fibronectin, the various things that constitute the extracellular matrix of our tissues. So these focal adhesions couple the cytoskeleton directly to the external world. What happens to an actin network if it's tacked down in several spots? Well, assuredly, if you pick two of these spots, let's say this one and this one, or this one and this one, and watch over time, if there's a big cable of actin filaments and it engages with motors, then you'd expect to see, let's see, I'm going to click these two here, right? So if they start out this distance apart from each other, you expect to see them get closer together, right? As this bundle contracts. No, you don't. Because these are tacked down to the substrate. You wouldn't expect that kind of thing at all. Instead, you'd expect that they'd stay in a relatively fixed position attached to the substrate as they are. But yes, the filaments between them might become bundled because what starts out as a loose mesh like this, as motors engage between two attachment points, weaves some sort of yarn into a distinct bundle like that. This is a common enough feature of crawling cells that they show up over and over again, and we often call them stress fibers. You can even see the existence of stress fibers in cells on glass without necessarily staining for anything because they are big bundles of stuff. What do? Probably nothing. Again, if these cells are tacked down to the substrate through these adhesions, then they can develop all the tension they want there, organize a bunch of filaments, but they're not necessarily achieving much. So what's the point? In real tissues, we're not talking about glass that they crawl on. Okay, some cells really do crawl on stiff substrates, almost glass-like. For example, the keratocytes that heal wounds in fish, they're crawling on fish scales, right? Those are a stiff substrate, not unlike a cover slip. At any rate, cells crawling in real tissues, they do experience a deformable extracellular matrix, at least to some extent. And in fact, the tension developed between sites of adhesions can itself organize the extracellular matrix into bundles of proteins and tracts that cells have made as they traverse them. Let's take a little closer look at this schematic of a stress fiber for a minute. So again, this is supposed to be something like a focal adhesion, a site where the cell establishes a physical coupling to the extracellular matrix, the ECM. So this is the plasma membrane of the cell here, spanned by something like integrins, these receptors for extracellular matrix molecules. The purple blobs are supposed to represent whatever, some proteins that then in turn couple the integrins to actin filaments, which is the red objects. And then the blue things looking like bones up there are supposed to be motors. Now, as it happens, focal adhesions are not necessarily just sites for filament capture. That is, they don't just latch on to actin filaments running around. They may be sites for filament assembly and nucleation. That is, around focal adhesions, there might be a lot of barbed ends, which would enable those motors to engage constructively and mediate contraction with those that happen to reach far enough to provide an anti-parallel, correctly oriented set of filaments for them to engage with. So you have this situation where there's an attachment point at the boundary of a bundle of contractile proteins with the motors concentrated more or less in the middle of them, right? And that starts to look a little bit like a muscle. Now, I mentioned, incidentally, stress fibers are a really common site in cells grown on glass, especially when they stop crawling around and start to feel a little hungry. They look like they could do something a little more constructive in a tissue where the extracellular matrix can actually deform, where the thing that they're crawling over is also soft material. In fact, if you plate crawling cells down on silicone sheets instead of on glass, if you put them on rubber sheets, you can see them wrinkling up the silicone sheets between adhesion sites. The only trouble is, in real tissues, people rarely spot stress fibers inside crawling cells or anything like them. So it may be that this represents an extreme case of a dynamic machine that has usually disassembled and moved on by the time it gets even halfway toward this kind of organizational state. At any rate, it looks sort of like the beginnings of a muscle. That is, a contractile bundle that's anchored at both ends with the motors in the middle between the attachment points. In fact, smooth muscle looks not unlike sort of an extended tapestry of stress fibers. But now let's take a close look at skeletal muscle sarcomeres, which I mentioned early on, and now we're going to look at their components in somewhat more detail. I'm going to draw the boundaries as a zigzag, just because they always call it a Z-line. These are the boundaries of the sarcomere. I guess I'll write that down here. The fundamental contractile unit in muscle. A myofibril, the contractile bundles within a muscle cell, a skeletal muscle cell, consists of many, many sarcomeres lined up in a row. The sarcomere is typically a couple of microns long from here to here, maybe 2.5, something like that. They vary depending on the muscle, the species, etc. I'm not going to draw every filament and every motor in the sarcomere. Instead, I'm going to draw a few and decorate them with some key components. The actin filaments do not span the entire sarcomere. Actin filaments are embedded in the Z line, and I'm going to draw them reaching, maybe 2/5 of the way across, something like this. So let's have a few little actin filaments in here. Actin filaments are arranged in an almost crystalline array within the sarcomere with a very regular spacing, and the so-called thick filaments, if actin is the thin filaments, the thick filaments are arranged amongst them. These are muscle myosins. Muscle myosin is enormous. It is an enormous bundle of myosin proteins. And I am not going to draw all of the heads that might be sticking off this bundle to engage with actin filaments, because a single one of these units, a single one of these bundles might have 300 heads or so. Lots and lots of heads to engage with how many actin filaments? Well, if they're stuffed in there between a bunch of actin filaments like this, you can imagine that there might be, say, 3, 4 actin filaments within reach of this bundle. So very many heads are engaging with each actin filament during contraction. How come the actin filaments end where they do, or start where they do for that matter? Let's see. All of the barbed ends of these actin filaments are back here in the Z line. So they grew by addition of monomers near the Z line. They grew and were sort of extruded back that way as they grew. And they are now capped, each of them, by a protein associated with the Z line. This yellow cap that I'm drawing in is called cap Z, because it's a cap and it's in the Z line. How come they only got this long? Well, that's a funny story. The pointed ends, they're capped by another protein. This one is does not have quite so intuitive a name. This is a little protein called tropomodulin. And it keeps the pointed ends from shrinking or growing, but that doesn't explain how they have a particular length and why they all end here. Does that have something to do with the myosin, perhaps? No, it doesn't. I'll draw another one in here. Let's give it its own cap Z and extend it out. Oops, a little thicker would be nice. The so-called ruler hypothesis says that a very long protein, nebulin, measures this distance and determines where this cap is placed, and thus how long the thin filaments, the actin filaments in the muscle will be. So this purple thing here, that's nebulin. But what about the sarcomere as a whole? This line here is known as the M line. I assume M is for myosin. Nebulin only says how long the actin filaments get. What about the entire sarcomere? Well, there's another protein, another giant protein. I guess I'm going to draw it down here. And I'm drawing this one as a spring, except I want a slightly different, better green chalk that shows up. I'm drawing this protein as a spring, at least for part of its length. This is another giant protein called titin, because it's titanic, get it? The idea is titin determines the overall length of the sarcomere and also provides it with an elastic component that limits both how much that sarcomere can stretch and also helps it not to get torn during contraction. The maximum contraction length, in turn, is set by the relative ratios of the myosin in there and by this myosin filament and the length of the actin filaments. In some muscles, like insect flight muscle, that's quite short. They only contract by a few percent at most. In others, it's much longer. Now, I've left out a really important component, and I need to find another color of chalk for it. I guess white is going to have to do. Wound around every actin filament is a long protein It's not one where the entire protein spans the length, like nebulin. This protein is called tropomyosin, for what it's worth. Tropomyosin is decorated with a little helper, a little friend. I guess it's going to have to go in brown. There's actually a couple of them in here. So these brown objects are yet another protein. I better complete this one because tropomodulin's function is to prevent myosin from engaging with the actin filament. Wait, what? Really? That's right. Until the signal comes along, all of those myosin heads in there are prevented from binding the actin filaments by the presence of this long, thin succession of tropomyosin proteins that are basically lying over their attachment sites until these little brown blobs, those are the troponins. Somebody really liked the tropo word here, right? In response to calcium, the troponins yank tropomyosin out of the way and let myosin interact with actin and therefore contract the sarcomere. So now that they're all drawn, let me go back and review and number these key components. The overall length of the sarcomere, and also its resistance to just coming apart when other things pull against it, like the neighboring sarcomeres, is due to this large elastic protein, titin. Of course, let me not forget to point out actin itself and myosin. I guess those should have been number 1 and 2. Then number 4 here, tropomyosin, prevents myosin and actin from interacting until the right signal comes along. Nebulin, number 5, is this molecular ruler that sets the length of the actin filaments, which in turn has an effect, of course, on the extent to which this muscle can contract, but that's a complicated feature. And then finally, number 6 is the existence of these capping proteins. So tropomodulin here gets a number 6 as well. These capping proteins stabilize this structure so that skeletal muscle is in some ways entirely unlike other actin-based superstructures that we've discussed. It is not a dynamic assembly. It is not continually turning over. Okay, there is actually some exchange, and a lot of the exchange in skeletal muscle turns out to happen at these pointed ends. Tropomodulin is coming off every once in a while, and some monomers exchange at the ends. So muscle is not totally static, but it is not a constantly rebuilding assemblage the way, say, the actin network of the lamellipod in a crawling cell is. Muscle cells build this assembly, and then it works over and over and over again with minor repairs. But how does it contract? So let's finish the story there. If this is the plasma membrane of a muscle cell, or a section of it, the plasma membrane undergoes an action potential in response to the neurotransmitter delivered at the neuromuscular junction. That is, the voltage of the muscle cell membrane changes in response to acetylcholine, as it happens, delivered at the synapse. That electrical potential is conducted deep into the cytoplasm by tubes called T-tubules. They act like little wires that conduct the voltage change down into the cytoplasm. The myofibrils, we'll imagine we're cutting along a cross-section of a muscle, and these myofibrils are, imagine now, we're seeing all those actin filaments arranged in cross-section in this crystalline array. I'm not going to draw them all in there. So these myofibrils deep in the cytoplasm are surrounded by sacs of the endomembrane system, a particular subcompartment known as the sarcoplasmic reticulum, in which calcium is sequestered. So this sarcoplasmic reticulum, full of calcium, is a subdomain of the endoplasmic reticulum, and it's closely juxtaposed to the T tubules that extend deep into the cytoplasm. The final missing ingredient is that embedded within the sarcoplasmic reticulum is a voltage-sensitive calcium pump, or calcium channel, excuse me. So the T tubules carry the action potential deep into the cytoplasm. They run right next to the sarcoplasmic reticulum that wraps each myofibril. In response to electrical potential change along the T tubule, voltage-sensitive calcium channels in the sarcoplasmic reticulum open up and they dump calcium into the cytoplasm, surrounding the myofibril. Within the myofibril, then, troponin pulls tropomodulin out of its groove, lets myosin engage with actin, and contraction ensues.